we go. All right, let's pray, shall we? Heavenly Father, we thank you for the gift of this day. We bless and praise you that you are with us, that your word stands firm in the heavens and shall not be moved ever. Help us as your dear children to wrap our arms around your word, to hear what you have to say to us, and to figure out some ways to engage the people around us who are simply not hearing it, but maybe open to hear it. We ask this in Jesus' name. Amen. So we're kind of chasing this idea of let God be true, right? And again, this concept that let God be true, though everyone were a liar, as it is written, that you may be justified in your words and prevail when you are judged. Not to win arguments, rather that we can have conversations with people so we can encourage one another in the truth as God's people, and so that also we might be able to engage those who don't know Jesus as Savior and Lord. So I will mention this too, and this isn't online, Roberta, but someday I can get you one. This Why the World Looks So Young kind of goes with last week, if you will. And it's just a little booklet that has a whole bunch of different facts in it. So it's just there for your use and growth and learning. We're chasing the origin of the universe and life. So last week we looked at the bigger, how did the universe itself come into existence? We looked at some of the geology. We looked at some of that stuff. Today we're going to chase, can non-living matter come alive? The short answer, of course, is no. We're going to look at DNA, and we're going to look at that as an information system. We're going to look at things like natural selection and mutations and all of that kind of stuff, as well as biblical kinds. So as we kind of get into that, in Genesis chapter 1, we find the words, then God said, let us make man in our image, after our likeness, and let them have dominion over the fish of the sea and over the birds and of the heavens and over the livestock and over the earth and over every creeping thing that creeps on the ground. So God created man in his own image. In the image of God, he created him, male and female, he created them. There's a claim here. And the claim here is that we are a beloved creation of God. That he made us specifically. And as you read through Genesis 1, God says, let there be and there was. 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 When we get to this point, he says, let us make. There's a personal involvement in our creation. And as we think about that, the big question here that we're wrestling with in this area, are we truly a beloved creation of God, or, as the culture insists, simply a random accident? And it's curious to me because if we're a random accident, the people who hold that view get very upset if you challenge that view. It's like, well, if we're all random accidents, right? And the conversation we're having is simply a result of the interaction of lifeless chemicals that have somehow animated themselves, that mean nothing, have no purpose, have no future, have no destiny, then why are you so upset? Why are you so upset about the fact that I believe that that's not true? It's curious to me. There's a logical disconnect here, right? I've heard it said this way, atheists say there is no God and they hate him. It's kind of like, folks, you can't have it both ways. So as we kind of roll through this, Psalm 139, you formed me In my mother's womb, you knitted me together. In my mother's womb, before my inward parts, you knitted me together. I praise you, for I am fearfully and wonderfully made. Wonderful are your works. My soul knows it very well. Your eyes saw my unformed substance. In your book were written, every one of them, the days that were formed for me, when as yet there were none of them. That ultimately God knows the span of our life, and recognizing he's not controlling the span of our life, if you will, in the sense that, oh, now you're going to do this, now you're going to do that. But for him, all time is now. So he sees us in our entire history, right? Now. I don't get how that works. But for him, all time is now. And all of my days are recorded, which means there's purpose, there is meaning, there is his value put into my life. So as we think about this, I want us to look at some things, the wonder of God's creation, human life. And there's a lot of stuff that we could be looking at. But as we get into this, we're going to start here. And ultimately, what this is going to show us is something that is happening in every cell. Well, maybe not some of the really specialized cells, but basically in every cell of our body as we sit here. And it's an animation. And I will say this, as complicated as this looks, it's simplified.
This animation demonstrates how the digital information encoded within DNA is used to direct protein synthesis. This is a DNA double helix containing the digital code which directs the cell in all aspects of operation. And here we see a protein complex called an RNA polymerase traveling down the DNA strand. As it moves down the strand, it carefully unwinds the DNA, preparing it for transcription. Inside the polymerase, we see a single-stranded copy of the original instructions being assembled as individual bases are positioned and added to the growing strand. A stop code marks the end of the protein specification, at which point this copy, known as a messenger RNA transcript, exits the polymerase and heads towards a two-part chemical manufacturing machine called the ribosome. While the messenger RNA moves towards the ribosome, transfer RNA molecules attach to specific amino acids in preparation for assembly. As the messenger RNA transcript passes through the ribosome, the process of translation begins. Using the instructions encoded on the messenger RNA as a template, the transfer RNA molecules align specific sequences of bases to corresponding amino acids, creating a protein chain. As this chain exits the ribosome, it is met by chaperones which prevent premature folding, while escorting the protein to a barrel-shaped machine called a chaperoning. This machine helps fold the protein into the precise shape required to perform its function. Although it is unclear how the chaperoning achieves this, we do know that accurate folding is essential in order for the protein to accomplish its intended function. Once the protein is complete, it is released into the cytoplasm to do its job.
Okay. No. You can stop now. Here we go. Whoops. No. All right, let me try it this way. Okay. What you've seen there, again, is simplified. The DNA is a double helix. As it's unwound, if you've ever played with a string or a rope that's multiple strands, and you pull it apart in the middle and you start pulling, pretty soon it's stuck. You know what I'm talking about? So there's literally another machine that follows that cuts one strand of the DNA and then resplices it so it doesn't get all tangled up. And it showed the polymerase running down the DNA, and just this a messenger RNA just floating out there in space. That doesn't happen. There are other structures that grab that. They take it to a nuclear pore because the DNA is in the nucleus of the cell. They open that pore, they send it through to other machines on the other side that then take it to the ribosome, you know, and they show it coming out of the chaperone and all nicely folded, which it does. What they don't show is all the other little molecular machines on the inside that take it to the place in the cell where it belongs, so it can do its job. With me? Yeah, I got it, right. I'm not with me either, okay? Incredibly complex. So let's take a look at another little thing. And this does go on pretty much in every cell in the body. We know that if we don't eat food, we die. We know we need food for energy, okay? But the blueberries I ate this morning don't show up in my cells as a blueberry, okay? They ultimately run through a whole series of chemical reactions, and what they really show up at is ATP, which has to be manufactured from all that stuff. All right. And that whole system is way more complex than my little brain can understand. But here's the whole ATP piece. Oops.
This animated sequence shows the ATP synthase enzyme in operation. The animation is based on an incredible series of scientific discoveries. Only the colors show artistic license. ATP, or adenosine triphosphate, is the energy currency of the cell. ATP is produced by a tiny molecular rotary motor, rotating at up to 7,000 RPM. These are so small that 100,000 would fit side by side in a millimeter. A current of protons drives the motor, unlike man-made electric motors which use electrons. This portion of the enzyme is where adenosine diphosphate is combined with a phosphate ion in the presence of a catalyst to produce ATP, which is then released, making way for the next cycle. A top view of the enzyme shows the sequential operation. Almost every biochemical process in your body requires ATP. Such a nanomachine exhibits all the characteristics of super-intelligent design. ATP is vital for life, and many of these motors were needed before the first living cell could exist. An evolutionary impossibility.
So, whoops, we got to cancel that. I did this earlier. I didn't have to cancel those. ATP, energy for life. So here is the dilemma that we'll look at as we go on. The instructions for building an ATP synthase are in the DNA. But you need the ATP to power the machines to get the instructions off the DNA and build it. You follow what I'm saying? So you can't build it unless you have ATP, but the instructions to build the machine that creates the ATP are round and round you go. It's all got to be there. The enzyme increases the reaction time tremendously. That's what enzymes do. If you've heard the word catalyst, right? Catalyst is something that causes a reaction, but doesn't itself change, but it speeds the reaction up. That's basically what the enzymes do. They have found enzymes that literally increase the speed of reactions by 100,000, a million, a billion times what it normally would have been. And all of that stuff is operating inside me and inside you. So let's take a look at one more here. And this is kind of a crazy little deal here. Remember, those proteins have to be delivered to somewhere within the cell where they're going to do their job. And this is how that happens.
Inside a living cell is an amazing transportation system. Proteins have to be delivered to the correct part of the cell to perform their intended functions. This animation, based on a lot of clever research over a number of years, shows how it happens. Highways made of microtubules are assembled by interlocking proteins, each manufactured in accordance with the coded instructions on the cell's DNA. Marching along a microtubule is the kinesin motor, the hero of our story, carrying a huge sack of proteins to be delivered to a predetermined place in the cell. Here, the proteins will be released to fulfill their functions. A kinesin linear motor uses one ATP to provide the energy for each step and takes 125,000 steps to cover 1 millimeter. This amazing machine shows all the hallmarks of design.
So the huge kind of unspoken deal in microbiology is that our living cells are so incredibly complex as to defy imagination. It literally is done that way. And as we look at some of these things, it's kind of like, here is the wonder, right, of what's going on inside of all of us, okay? And let me see here, for some reason you did not go back down where you belong. It's amazing what's happening. In that packet, there's a single sheet that says 15 loopholes in the evolutionary theory of the origin of life summary. A lot of this is somewhat complex. I'll point out a couple of things, though, on this piece. Ultimately, you may have heard of the Miller-Urey experiment somewhere in your science, where they put a bunch of chemicals together and sent lightning in there and collected amino acids. Anybody does that recall? Ring any bells in biology? Oh, maybe they've stopped teaching it since it's everything but correct. The thing is, okay, well, you probably are, in our education system, you're probably ahead in that exchange, right? And let me be clear, actual observational science, which is what all these little videos were based on, is awesome. It allows us to do incredible things in life that otherwise we couldn't do. Historical science, which is like, here's how everything came together, is garbage. And unfortunately, what's happened in our culture is the word science has been defined as both of those things when we really are talking two different things here. So in number one here, basically it says that what was used in the famous experiment, that atmosphere didn't exist. If we skip up to number two, we find out that there was oxygen in that early atmosphere. Actually, it wasn't as early as they think it was, but there was oxygen there, which destroys biochemical or organic compounds. Number four, same kind of thing, all of the energy sources that would produce biochemicals destroy them faster than they can make them. So to get life from non-life, you have to build these biochemicals, right? And we're going to look at that in a moment, just what exactly are we talking about there. If we go to the back of this sheet, number 10, long periods of time do not help evolutionary theory if biochemicals are destroyed faster than they're formed. And everything in the research says, yeah, if you've got basically amino acids, we've been told are the building blocks of life, but basically amino acids are chemicals. They do not live in and of themselves. When they're properly built into proteins inside a functioning cell, then they do things that cause, that keep us alive, if you will. But basically, they're just chemicals. And long periods of time don't help you if the biochemicals you need to randomly build are destroyed faster than you can make them. So ultimately, we'll talk about where evolutionists are at with some of that. let's go a little different direction here. This is Louis Pasteur. You may have heard of him. He did a very famous experiment because in Lewis's day, they thought that life just spontaneously arose from matter. Now, how did they come to this conclusion? They came to this conclusion because you could take something, for instance, like a meat broth and stick it out, and pretty soon it was growing things, right? We know how that works. You know, it's like you drop that piece of food and it rolls up under the counter where you don't see it, and pretty soon you're like, what am I smelling around here? I don't know. And you find it's a science experiment. Look. Yeah. What he did is he took a beaker and he boiled it with that S-shaped curve on it, so that he drove all of the, basically everything out of there, and then sealed it, and he had no growth. When he would break the neck and wait, then he would have growth, which proved, hey, non-living things don't bring forth life. There had to be something that they couldn't see creating the molds and so forth and so on. And this law biogenesis, says life comes from life, period. And as far as we know, it has never been violated. So every time you read the paper, oh, wow, scientists made this discovery. We figured out how non-living things become life. Read a little further on and, you'll find a lot of conditional language. Perhaps, maybe, a step in the right direction. And it's like when you get to the bottom line, it's like, nope, the law of biogenesis has not been disproved. So if we think about the complexity of life and how difficult it is to get life from non-life, proteins are the backbone of cellular function, right? Proteins are made in the cytoplasm of the cell. That's not the nucleus, that's out here. However, the instructions for the proteins are found in the DNA in the cell nucleus. So the nucleus has its own little membrane, cytoplasm is beyond that, and that has its own little membrane out there, and then all those cells are joined together beyond that. But basically, you've got the instructions in the DNA, but it's manufactured out in the larger cell. Now, here's where it gets incredible. You've got a whole set of molecular machines, and I use that term because actually that is a term that's often used. I mean, we saw some pictures of that, the little kinesin motor, right? I mean, you look at this thing, it's like, dude, I want one as a pet. They're just so cute. They're just walking right, one step at a time kind of a thing. It literally is a group of proteins that is built for that function. And somewhere, not only is it built for that function, but built into it is information to walk down this microtubule to this point in the cell to deliver the proteins that it's carrying. It's incredible to me, the complexity of this. So molecular machines retrieve instructions from the DNA. They transfer these instructions to other molecular machines in the cytoplasm that gather the needed materials, build and fold the protein. And by the way, it's a 3D folding. It's three-dimensional. So I can have a whole string of amino acids in the correct order, and if it's folded wrong, it doesn't work. The curiosity about a string of amino acids is it wants to self-fold. And if it self-folds, it's going to be wrong. The complexity just boggles my mind. It just boggles my mind. All right? And then it delivers the protein to the correct location in the cell for the protein to function. Enzymes are chemicals, basically, that speed this process up. And I gave you a little piece here, and again, there's so much information out there, it's incredible. World record enzymes, and basically what it talks about here is some things that they have actually discovered that, hey, this thing increases it by a rate of 10 to the 18th power. What the heck does that mean? It's a 1 followed by 18 zeros. That sounds like it's fast. There's one piece on the back here. Well, on the front, there's kind of a little cutout over here on the side that says, without catalysts, there would be no life at all. Period. From microbes to humans. And somehow, if all of this just happened, you have to explain how the enzymes came into existence before the process they were supposed to speed up existed. Yes, an explosion did it, because we know explosions, we know that if we take a big bomb out to a junkyard, we can assemble a 747. Exactly. No problem. Facts, those nasty little facts. Let's just get rid of all of them.
Don't need to worry about that.
Yes. Well, that's true. I mean, my daughter has named her car. It's just, his name is Kevin. Don't ask me why. Okay. It just happens. Anyway, there's an update on the back here. This particular researcher, Dr. Wolfenden, has co-authored a paper on an enzyme that speeds up a reaction that otherwise would take 2.3 billion years. And it's just a small little reaction that helps build hemoglobin and chlorophyll. We don't need those, do we? Chlorophyll is the green stuff in plants. The whole life cycle is built on, and hemoglobin gets the oxygen to our cells so we don't just all fall over, right? So it's just tiny little things like that. So in some respects, it's more complicated, and this is on the handout on page 2. It's more complex than that brief description. No DNA, no life. No molecular machines, no life. No proteins, no life. No enzymes, no life. Instructions to build molecular machines are found in the DNA. Instructions for the proteins are found in the DNA. Instructions for the enzymes are found in the DNA. Are we getting to the point where we believe that DNA is the most sophisticated information management system known to man? And very little is really known about its true complexity. There's a geneticist by the name of Robert Carter who has done a lot of study on this. He works for Creation Ministries International, and he describes DNA in a 4-fold description. If you go way back to biology when you were talking about baseball, and you remember on cell division how all the chromosomes line up, right? They double and they line up before the cell divides, and they're all in a beautiful array. That's, basically your first dimension. All of these things are out here. Now, they're not doing anything for cellular function at that point to speak of, because they're not interacting with each other. So they pull apart to create new cells. Well, the second piece is once it's actually in the cell, those chromosomes start twisting together. And this chromosome will come next to that chromosome because the cellular function requires instructions from both. And you've got all of this as they interact and they create different proteins, right? That works. The third kind of function of this is that the shape changes in the nucleus. The DNA literally changes shape depending on what the body needs. And the fourth level of this thing is that it changes over time. And remember when they said if you eat a good diet, it's healthy? It literally is healthy at a level of DNA. They have done some studies. They're not fully conclusive because this thing is so blasted complex. But it's like if you eat certain foods, certain genes that tend to do good things for you turn on, and certain gene combinations that do bad things for you turn off. And if you eat other foods, the opposite is true. They've done some research in diabetes. That if you have certain things in your diet, you're not as likely to develop the disease, although you're genetically predisposed to do it. If you eat other things, you're more likely to develop it. And it's like, I don't get it. Okay? I don't get it. The guy got his PhD by basically, I don't remember what it was. It was like a blue fish he turned green or a green fish he turned blue or something by playing around with the DNA. That's how he got his PhD.
So would that mean okay, so I'm diabetic, and so would that mean like if we have kids and we don't let the kid have sugar, they have less of a chance of having diabetes?
It's a little more complicated than that, but essentially, yes.
That's kind of what I thought we were going.
Essentially, that the food that we take in can alter the expression of our DNA. All right. Now, it's not absolute. It's like if you absolutely follow this particular plan, nothing's going to happen.
Nothing's perfect.
But the thing is they have found in their research, and part of it because so many people have that disease, there's a lot of research going on, and they've said, hey, you know, if we do this and this and this, then all of a sudden, when they started doing the genetic studies, they're going, hey, it turned that set of genes off and it turned this set of genes on, and they're doing that. And, you know, proving causation in a system this complex, good luck. Just good luck, right? We're also talking about something here called irreducible complexity, and it's a whole lot easier to explain with something like this, right? So you have a whole set of pieces here. You have a base, you've got a spring, you've got the trap wire, you've got a trap wire holder, you've got a trigger, and you've got bait. The question is, does the mousetrap work if you pull any piece out?
You get rid of the bait if you had a really, really stupid mouse.
Okay, so let's take the bait out of the equation. If we take anything else out of the mousetrap, does it work?
The trigger. If
Well-
you just set the bait where the trigger's supposed to be.
Well, but the thing is the trigger is what, you know, that trap wire is spring loaded. If that trigger wire is not hooked into something, it's going to be sprung, right? And the idea of irreducible complexity is you have a system, you know, that needs all of the pieces to function. Now, you know, let's take it to your automobile engine. I mean, you can open up the hood and say, I'm just taking that piece out right there. Now, some pieces you could probably take out and the thing would still run. Other pieces you take out, you're done. Biologically, the understanding here in terms of irreducible complexity is this, is ultimately DNA cannot replicate or build anything. The molecular machines that the DNA has the instructions for are required to carry that out. And the system is irreducibly complex. Take out any piece and it's lifeless food. The other piece is this, which is just fascinating to me. At this point in time, as near as I've been able to figure out, looking at different sources, the DNA contains coding regions for proteins. It contains 20 to 25,000 regions that say, here's how you build that protein. But in a human body, you have anywhere from 80,000 to 400,000 proteins. So 25,000 instruction sets make 80,000 to 400,000 proteins. And the non-coding DNA is what controls that. Because the non-coding DNA will say, take this stretch here, go over here, take that stretch there, come over here, take this stretch here, and build that protein. They have discovered sections of the DNA where if it's read in one direction, you get one protein. If it's read in the opposite direction, you get a completely different protein. There's no human information system known to man that you can do that. Right? Now, there are some English palindromes, you know, Bob, you spell it forwards, it's Bob, you spell it backwards, it's Bob. But when it comes to amino acids, it's like God and dog. It's like, nope, it's not the same thing. Right? And ultimately, this is what happens here. Let's dig in a little deeper and figure out the chances. So I've kind of listed this for you on page 3. we're going to build a protein of 150 amino acids. That's actually a relatively modest protein. The average length of a protein in the human body is 300 to 350 to 400 amino acids, and some of them are just ginormous. You know, literally thousands of amino acids put together. But we're going to build a little modest one. Now, there are 20 different amino acids from which life is built. So, and they will all connect to each other. So we have a 1 out of 2. If we're going to randomly do this, let's say we got the amino acids just floating around in whatever it is, and we've got all of them there in whatever quantities we need. But that means at any one of those positions, you have a 1 out of 20 chance of getting the right one. Does that make sense? You've got 1 to 20, and 1 to 20 can hit any one of those 150 spots. But to build the protein, you have to have the correct one in each spot. So the random chance there is a 1 out of 1 followed by 74 zeros. Huh. It gets better. It gets better, okay? All the bonds have to be peptide bonds, not amide bonds. So there you have a 1 out of 2 chance at each one of those bonding positions. You've got 149 bonding positions in there. That's, you know, 1 out of 1 followed by 45 zeros. Hey, that's a much better chance, right? Now, curiously, and you'll see that in this little impact article, it says impact 371. And one of the things that we need to know about amino acids, if you look at this little diagram in the back here, or on the back of page 1, you'll notice that you've got mirror images there. Amino acids come in mirror images, L form and D form, L for left and D for right. Why it's D for right, I don't know. Somebody could explain that to me someday or not. But the issue is if I'm going to build a properly functioning protein that's going to fold correctly, they all have to be L form. So I'm back to another 10 to the 45th power. Now, if I add all of this up for this modest little protein, okay, I have 1 chance out of a 1 followed by 164 zeros. Eyes glazed over yet? By way of comparison, it is estimated that the number of electrons in the known universe is a 1 followed by 80 zeros. So the chances of building even one of these tiny little proteins is more than double the number of electrons in the universe. But we're not done yet. Okay. If we have one protein formed by random chance and it's folded properly, which is another whole issue, you still don't have life. The simplest one-celled creatures today have about 2,000 proteins. So if we look at that number, it's just beyond imagination. It's a 1 followed by 328,000 zeros.
So you're saying there's a chance?
There is a chance. There is a chance. Yes, there is. Yes, there is. We're going to get to that in a minute. Okay. Now, evolutionists have hypothesized that you could get a living cell with only 250 proteins. Now, they haven't actually fully described what those 250 proteins are and what they actually do, but that's their theory. So let's be generous. Now you have a 1 followed by 41,000 zeros, a much better chance than 1 followed by 328,000 zeros. But recognize, if I am doing statistics, anything more than 1 in a billion is considered non-random. That's 1 followed by 9 zeros. And we're in the big numbers so big they don't have names. All right? And then on top of that, it's got all of these things would have to be folded correctly, which in our cells require a very specific molecular machine. So basically, molecules to man is in science, it's fake. It really is. molecules to man has to ignore clear and compelling science to hold on to it. No matter how much time is involved, non-living matter does not come alive. So the question is this: leading evolutionists today who say evolution is an established fact of science, all of science is built on this established fact, the louder they scream, the more you should doubt what they're saying, right? When you really pin them down and ask them, how did life evolve? How did it go? How did it happen? E.T. did it. I'm not kidding. I am not kidding. Okay? And there are two different ways. One is direct interaction. E.T. showed up, looked at the planet, said, oh, look, it's a planet that's got primordial soup. Zap. Good. Our job is done. We'll go on and find the next place. All right? The other way that is a popular theory is called panspermia, which means somehow you pack this biological material into asteroids, send it through deep space in deep time, and an asteroid came through the atmosphere, was not incinerated completely on the way down, fell into a pile of whatever it was, and presto changeo, here's life. It is. It's ridiculous. It would probably make a really good book series. It probably would. I think you're probably correct. So, yeah, as usual, I am.
I can't remember the name of it, but basically we were an alien experiment.
Right.
And then
that went awry.
no, it didn't go awry actually, because their time worked differently than ours, so they were controlling it the whole time to see how it went. They were basically supposed to stop back and check up on us every now and then, except something went wrong with some of their technology and they came back to us. And when they finally did, a lot of really interesting things have happened, like most of modern history. And there was also a virus in this book that was killing off everybody. And the aliens had to stop this virus because it was ruining their experiment.
Sounds like a good thing to put next to your standard biological textbook. So one of the things that we're going to think about here, and I'll show you her picture in a minute after we look at this. On page 4, I've kind of got a summary. She's going to talk about mitochondrial DNA in time. And I want you to kind of walk with her to the conclusion that she arrives at. So this goes a little bit longer, but it's worth it, I think. And I just got to mind my minutes.
Does human mitochondrial DNA evidence support the biblical time frame? So what are we talking about here? Well, let's talk about some facts. First of all, mitochondria are the powerhouses of the cell. They're little tiny organelles inside our cells that produce energy. They're very important for that. They're inherited maternally, so we get those from our mother, not our father. It has a circular genome, which you see there, of about 16,000 base pairs. So they have their own genome. Now, genome basically means the entire sequence of DNA, the information in that. You have a nuclear genome, which is what most people are familiar with, but you also have a genome inside each of those mitochondria. And it encodes several genes, which are important in what the mitochondria and what goes on there, as well as some regulatory region called the D loop that controls those genes. So that's just some basic facts about the mitochondria itself. Now, how is this related to the issue of a biblical time frame? Well, for that, we have to go to the mitochondrial DNA clock, a molecular clock. And don't worry, we're going to talk about that. And mitochondrial Eve, which probably a lot of you are familiar with. So first of all, let's talk about what is a molecular clock. Well, basically, it relates the time that two species diverge to the number of differences between their DNA sequences. So, for example, the greater the time there is since two species diverge, the more differences you should see because they've been on their own path for a longer period of time. But the lesser time since they've diverged, if this is a short time, there should be much more similar. There should be less differences. Now, something you need to know about molecular clocks, they are not an independent measurement of time. They are based on a certain assumptions about the past. They are calibrated according to certain things. The main things that they are calibrated with is that basically assuming that the fossil record is accurate, radiometric dating is accurate, and that humans have diverged from chimps. Okay, so right away we can see there's going to be some major problems here with the assumptions they're using to sort of calibrate these clocks to figure out how fast this molecular clock ticks, so to speak. So that's what a molecular clock is. Now, what is mitochondrial Eve? Mitochondrial Eve is the maternal most recent common ancestor for all living humans. And she is believed to have lived around 100,000 to 200,000 years ago, based on the fossil record, radiometric dating, things like that. So basically, we all have her mitochondria. Now, this is in no way reference to the biblical Eve. The evolutionists for 1 minute do not believe there was one woman and we all have her mitochondria, but rather that one woman from a larger population of women, for whatever reason, it's her mitochondria that we all have today. And so I want to make that clear. Now, how do these two things relate? Mitochondrial DNA clock and mitochondrial Eve. Well, a man by the name of Parsons, a scientist back in 1997, published a paper where he looked at the control region, that regulatory region of mitochondrial DNA as a molecular clock. So he was going to use that to study some relationships. And what he expected to find was that there would be one mutation every 12,000 years. That's what he expected to find. Why did he expect to find that? Because based on the fossil record, based on radiometric dating, all of those things, that humans diverge from chimps, that's what they had already assumed the rate would be. But they found something very different. Now, what he was looking at was modern human DNA, mothers, daughters, grandmothers, granddaughters, looked at 327 different generational events and found that actually the mutation rate is much, much quicker. It's one mutation every 800 years. Okay, that's orders of magnitude faster than what they ever expected. Now, that in and of itself isn't so much of a problem, but it is a huge problem for them when you apply it to human evolution. Because the problem is that mitochondrial Eve becomes 6,000 years old.
Oops. Oops. Yes, indeedy. Big oops, right? Big oops. So as we kind of look at this, Dr. Georgia Purdom is her name. That's part of a longer video. She is another one of these incredibly brilliant people who just knows all this stuff, right? This is her life. And ultimately what we're looking at here is that it reduces Eve, mitochondrial Eve, to 6,000 years old. Huh. So, single cell to everything, let's assume somehow we can get a cell, even though we can't, right? And I'll mention this too, that in the packet, there's this thing that says Adam, Eve, Noah versus modern genetics. And this indicates that there is actually one Y chromosome that is shared by every male on the planet, although there are variations of it. And that while there is a mitochondrial Eve from whom all the mitochondrial came, there are three major strands, M, N, and R is what they're called. And curiously, all of the different variations, this is the out of Africa theory, the little picture on the front, but all of the major variations run through the Middle East. Can we say Mount Ararat? Interesting, just interesting. Anyway, so let's think about this. First, the word simple can't be applied to any kind of living cell because by any standard of comparison, the most complex things we've ever built as human beings is, like stone tools compared to the cell. Second, even things like simple bacteria, right? Which is incredibly complex, but when you get to human DNA, we have about 1,000 times as much information in the DNA as many bacteria do. Third, the process requires a massive increase in usable information. So if we have that single cell, if that alien asteroid landed in the primordial soup, whatever the heck that is, okay, and the single cell was there, that actually just like kind of magically turned into us. But to do that, we have to increase incredible amounts of information. All right. If we look at information theory, and there's a lot of stuff out there on this, how does information work? All information has to have a source. And actually, information says that that source is a mind. Period. If we find information, somewhere behind it, there's a mind. Or at least something that behaves like a mind, right? DNA is copied information, which means DNA has a source, which should be the end of the argument right there. However, the evolutionists will say, no, 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 no. If you have the single cell, we can get from the single cell to a human being, no problem. And basically, they use these two mechanisms, natural selection and mutations. In natural selection, what you have is a bigger population, and the part of that population that can reproduce more offspring, that survives. Is natural selection biblical? Absolutely. As a biblical creationist, I believe that it's true. But one of the things we need to know about natural selection is normally it's a loss of information. So let's consider dogs. We've all seen dogs, right? So let's say you have a pair of dogs that have medium length hair, which means they have the genetic information for short hair and long hair, and they're heterozygous, which means they've got both of those genetic characteristics, which is why they have medium hair. Now they can have offspring, and as they have offspring, they can have dogs with short hair, they can have dogs with medium hair, and they can have dogs with long hair. But the short-haired dogs, the only genetic information they now have is for short hair, because that's recessive, right? You got to have gene from mom and dad to get that trait. So let's say the climate changes and all of a sudden the average temperature goes up 47 degrees or whatever it is. That's probably a little much, right? The long-haired dogs, they're dead. Medium-haired dogs, they're next. Pretty soon you're just left with short-haired dogs. That's natural selection. Environment changed, they adapted, the short-haired dogs reproduce more offspring. Pretty soon you have no medium-length or long-haired dogs. The thing is now, let's say the climate reverts back. The short-haired dogs all die because they do not have the genetic information to create the long hair anymore. It's gone. That's how natural selection works. Variability and adaptability are built into the DNA. All right? Survival of the fittest or differential reproduction is another way that this is talked about. Now, you will hear evolutionists say things like, well, bacteria have evolved because they've evolved antibiotic resistance. Actually, that's been studied in a number of bacteria. And what that actually is if you look at any population of bacteria, they don't quite have the same error reducing methods in their DNA that we do. We have machines on the inside that look for errors in DNA and repair it. That's happening every moment of every day. And basically what they found is antibiotic resistant bacteria were in the general population before antibiotics existed. They didn't do real well because the reason they're antibiotic resistant is one essential function is broken in that particular bacteria. But they thrive in the presence of penicillin because all the competition gets killed. Woohoo! So, mutations. Mutations basically are copying errors in the DNA or some sort of error there. Most mutations are too small to be selected by natural selection. They're just really relatively minor. Unfortunately, many of them are harmful. Some of them are fatal. There is a little problem here, though. Even those said to be beneficial do not add new information. So they will point to things like, look, you have blind fish in caves. That's evolution. No, that's the loss of genetic information. Now, is it beneficial to the fish in the cave? Sure, because eyes in an environment where there is no light, they can get injured. They're pretty much using energy that you don't need to use, all that kind of stuff. So somewhere along the way, there was genetic information on how to build an eye in a fish lost, and it's beneficial. Pull that fish out of the cave, throw him in the pond on the surface, he's dead in a hurry. right? So basically, even the beneficial mutations that they point to ultimately don't do anything, and there is no known increase in information. They had Richard Dawkins, world famous evolutionist, doing a video interview, and he agreed to do this with a creationist organization. You know, if I was him, I wouldn't have, but he did. And he was asked on camera, can you cite one mutation that has been scientifically studied and has shown that it has led to an increase in information on subsequent generations in the DNA? In the video, he's silent for about 1.5 minutes. The narrator comes on during that time and says, actually, he asked for the camera to be turned off. This pause was longer than 10 minutes. And never had an answer because there isn't one. So ultimately, no increase in information, no increase in information. From single cell to human being, you have to increase the information at least 1,000-fold or more. And there's no mechanism for it, none. So ultimately, we've got two different things here. A single cell origin, that diagram on the right, basically, this is what evolution teaches. There was a single cell and it brought all this other kind of stuff. The one on the left is the creationist orchard of life. You have biblical kinds, created kinds. Now, do you have variation? Yes. The curiosity is everything they point to in evolution. Well, the bacteria evolved. Well, let's see. You started with bacteria, you ended with bacteria. You started with a dog and you ended with a dog. And ultimately, this is where things kind of go off the rails. If we look at what God says in a lot of this, and that's in the notes here, is basically according to its kind or according to their kinds or things like that appear 10 times in Genesis. The word for kind is bara, and actually the Creation Museum has invented a word, baramin, which is biblical kind. All right? So as we look at this, here's where we want to think about this. Ultimately, evolution is not turning bacteria into other bacteria, or dogs into dogs, or cats into cats. What is evolution? Evolution is turning the mold in your refrigerator into your goldfish. Then you want to take the goldfish and turn it into your puppy dog. And you want to do that in discrete steps that can survive and reproduce offspring. Oh, by the way, all of which are missing in the fossil record. Oops. If you're wondering at this point in time, absolutely. If you're wondering at this point in time,
Imagination.
you know, why anybody would believe this. Now, here is an actual true fact here, then we're going to have to wrap pretty quick. Many people who have been raised on this don't have any of this information.
That's what makes me wonder if they truly believe this or they just don't believe in God.
And it could be both of those things, or it could be, and again, when you talk to some of these people who do these presentations on a more regular basis, they say a lot of times you'll have a conversation and somebody will say, I never knew. And the minute that they get to the point where they never knew, then they're ready to reevaluate the whole I don't believe in God thing, right? Because this all said there must not be a God. But when you look at the other side of the information that they're given in biology class, either in high school or in college, and you go, wait a minute, this isn't making any sense, right? Then where did it come from? Well, if anything, the culture has done us this service, if you will, because God is still, in charge of everything. If their story falls apart, the only other one that's out there in the culture is biblical creation. So if this thing falls to pieces, and this is where we can begin to engage people, right? I did give you one on mutations called The Blind Gunman, which basically is playing off of a Richard Dawkins book called The Blind Watchmaker, where he says, hey, mutations build us all. And basically he gives you examples of mutations that kill you and said, it's not the blind watchmaker, it's the blind gunman. You know, he shows up and shoots you dead. This one, if you like satire on the origin of articles, because we all know articles don't have authors. Because if you read the article, you don't see the author in the article. You didn't see the author writing the article. Therefore, it doesn't. This is the guy, Jason Lyle, the guy that I got all the logic and logical fallacies from. He wrote this on a blog, and he had evolutionists going crazy coming after him. I gave you one of the responses on the back, but the others on the web are also highly entertaining. So, as we think about this as well, there are distinct gaps between different created kinds at the level of DNA, which is why you can't turn that slime into your puppy dog no matter how hard you try. All right? Kinds can turn into kinds. You can take a zebra and a horse and get a zorse, or a zonkey, you know, with a donkey, right? But you can't turn it into a luck. Too much information gap in the middle. So, thinking about response. What does God say about the origin of life, of human life? We've talked about that. What does God's word say about created kinds of plants and animals? And there aren't that many. And most of them, by the way, are small. We'll take a look at a little bit of that next week. What is the foundation of the deep time concept? That deep time flows all the way through this. Cosmology, origin of life, everything, right? And ultimately, we looked at that last week. What illogical fallacies are being used? All of these are in there. Equivocation is a big one. The bacteria has evolved, therefore evolution is true. Well, you're talking about two completely different concepts using the same word. That is a logical fallacy. All right. Ad hominem, you know, faulty appeal to authority, the straw man. They set up creationist positions that are absolutely ridiculous. It's like, we don't believe that. I would knock that down. I don't believe that. Right? But you'll find that all the way through. And I've kind of given you examples of some of these in the notes. We really do need to respect the person. And again, I think in many cases, people they got into junior high and they were exposed to biological evolution. And it's like, okay, the teacher tells me this is true. Everything I'm reading says this is true. I go to the museum, it says it's true, da 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 da. Well, if this is true, because most junior high kids are smart enough to figure out if this is true, then the Bible isn't. Right? And so they give up at that point. And then their ears are hardened at that point. So we have to listen well and be compassionate, define terms, and this last thing is educate people with actual facts. Some of those little videos on what goes on in the cell, I find that stuff fascinating. Absolutely fascinating. And you telling me this just happened all by itself? And this is one of 1,000 processes going on in every cell in your body right now? Often makes people stop and think. With a head person, a heart person, a hands person, with the head, we really want to engage the larger things, don't get dragged in, but the complexity of life, the discoveries of microbiology, science has defined itself only in the material realm, but things like logic, purpose or meaning in life, love are immaterial. Where does it come from? Information theory is a good way to go after folks here. In the heart person, if we really live in a random universe, there is no hope. There is no purpose. There is no meaning. And for a heart person, that's intolerable. Right? So you approach it through that venue. With a hands person, if we really want to make a difference, we have to take action built on reality. Right? And actually, in some respects, evolution has gotten in the way of some science because all these assumptions come into it and they can't see what's right in front of them. So the other piece of this that we want to think about here is the culture tells this big story about how we've evolved. And one of the parts of that story is we're going to be bigger. We're going to be better. Everything is getting bigger and better, right? Bigger, better, bigger, better, bigger, better. Really? Is it? Is it? And yet, if one stays true to that evolutionary worldview, it's all irrelevant. What we really need to point people to is this love story of God, and we can point to the real bigger and better, that's heaven. That we have that abundant life now, but coming is something that is so much beyond us that it doesn't even compare. And ultimately, one of the things that I see in the culture is this idea that if we only do this, we can build a utopia, which is another way of saying we can build heaven on earth, except many people would be horrified if you pointed that out to them, because they don't believe God, right? There is no heaven, there is no hell, etc., etc. There's just this life. So ultimately, we've been down that road. Next week, we get to play with dinosaurs. Yay. Why? Major Trojan horse for children this tall and above, right? Because they get hooked into dinosaurs, and everybody knows they lived 65 million years ago. Because that's what the press release is all about. We'll take a look at some of that. So, blessings. Thanks for being here. Thanks for hanging in here with me. You can tell I'm passionate about this. I probably know enough stuff to be dangerous to myself and others in respect. But hopefully there's some things there that will be helpful to you. And again, I think especially in this area, if we can begin to engage people with questions about, hey, are you aware of this? Are you aware of that? Have you ever seen what's going on inside the cells in your body? That it will begin to raise the questions for you. Does that make sense? And don't assume that even, my son works with a group of engineers. All right. And he has some of these conversations as circumstances allow. And you got, these guys are brilliantly scientifically trained. Yes, as engineers. Calculus has very little to do with biology. All right. All of the stuff that they're doing is observational science all the way down the line. Because he works for a company that builds jet engines. It's mechanical engineering. It's electrical engineering. If they're doing chemical engineering, it's on fuels. It's not biology. So the reality is that they just accepted it because it was part of their training. But they've never really critically examined it. And if they use their abilities as a scientist to critically examine it, pretty soon they go, this is crap. You design a jet engine this way, baby. It ain't even getting off the ground. And if it does, it's not staying in the air. So ultimately, sometimes we can't assume that people know this stuff. So the Lord be with you and bless you this week. And I've got to roll. As late service awaits. All right, let me get back over here.